I think our slide is here. Okay, for your convenience, this slide provides the QR codes for our social media avenues. You can scan them now with your phones and be taken to the appropriate webinar, to the appropriate platform. Today's webinar in its entirety will run approximately 1 hour, which includes a question and answer session. And if you have questions now or during the webinar, please enter those questions into the Q&A chat pod. We'll answer as many as time allows. And now it's a pleasure to welcome our speakers. First, we'd like to welcome Dr. Vikram Arya, Associate Director for Therapeutic Review in the Division of Infectious Disease Pharmacology in the Office of Clinical Pharmacology within FDA's Center for Drug Evaluation and Research, or CDER. Second, we will hear from Dr. Brian Booth, Director of the Division of Cancer Pharmacology 1 in the CDER's Office of Clinical Pharmacology. And finally, we'll hear from Dr. Ethan Stier, Associate Director for Lifecycle Management within the immediate office in CDER's Office of Clinical Pharmacology. Please join me to welcome Dr. Arya.
Good morning, good afternoon, and good evening. It is my pleasure to welcome everyone to the webinar today on clinical pharmacology considerations for food effect studies, an overview of the FDA food effect guidance. My name is Vikram Arya, and I am an Associate Director for Therapeutic Review in the Division of Infectious Disease Pharmacology in the Office of Clinical Pharmacology in CDER, FDA. There are three broad learning objectives of today's discussion. The first is to understand why it is important to assess the effect of food on drugs. The second is to comprehend general considerations for designing food effect studies. And third, what are some data analysis considerations related to food effect assessment? Before we start the discussion, I would like to take a moment to define the scope of the presentation today. The left side of the slide shows the first page of the 2022 guidance on assessing the effect of food on drugs in INDs and NDAs clinical pharmacology considerations. The presentation today is based on the recommendations outlined in this guidance. During the presentation, I may also refer to this guidance as just the food effect guidance. What we will not discuss today are 1, the PhET bioequivalence studies, and 2, the PhET comparability studies. And the slide provides references to other guidances for obtaining further information. With that, let us review the roadmap for today's discussion. The presentation is divided into 4 major topics. As part of the first topic, we will discuss the importance of assessing the effect of food on the pharmacokinetics of a drug and how alterations in drug exposure due to food may have important safety and efficacy related implications. The second topic will focus on a few key recommendations for food effect assessment, such as the timing of conducting food effect studies during drug development, the idea of pilot studies to get an early read on the food effect, and food effect assessment related considerations during the formulation development process. As part of the third topic, we will discuss some general recommendations for food effect assessment, such as the trial design for food effect studies, what type of meals are recommended to be evaluated in food effect studies, and the concept of modified fasting conditions. As part of the fourth and the final topic, we will discuss some additional considerations related to food effect assessment, such as the application of model-informed drug development approaches for food effect assessment, evaluating food effect in specific populations, and data analysis considerations. Food can increase the systemic exposure of the drug, thereby potentially impacting safety and/or efficacy. Food can decrease the systemic exposure of the drug, thereby potentially impacting efficacy. And in some cases, the observed increase or decrease in the systemic exposure of a drug when given with food may not be clinically relevant. And in addition, there may be times when giving the drug with food is the only practical means of ensuring adequate systemic exposure. Changes in drug exposure, be it either increase or decrease in systemic exposure, are typically evaluated in the context of available exposure response information to determine what dosing instructions may be included in the label to ensure the safe and effective use of the drug. Food-mediated effects on bioavailability can potentially have significant drug development and regulatory implications. As we all know, food can have a significant effect on the physiology of the gastrointestinal tract, and some of the mechanisms through which food may impact bioavailability are listed on this slide. Food may impact gastric pH, thereby affecting the absorption of weak acids and weak bases. Food may delay the gastric emptying, or food may increase bile salt concentrations, which may impact the systemic exposure of poorly soluble drugs. Overall, the effect of food will likely be a combination of the physicochemical properties of the drug and the complex physiological changes induced by food. From a drug development and regulatory review standpoint, what we are really interested in is to address the following three important considerations. One, understand if systemic exposure is affected by food. Two, assess the effect of different meal types on the systemic exposure of the drug. And three, provide dosing instructions in relation to food intake. The next few slides outline a few key recommendations for food effect studies. The food effect guidance recommends evaluating the effect of a high-fat meal on the pharmacokinetics of a drug early in development. If a significant change in systemic exposure of the drug is observed with a high-fat meal, assessing food effect with a low-fat meal could be useful. Generally, the effect of food is initially assessed as part of the first-in-human single-dose trial, also known as pilot food effect assessment, to provide a preliminary assessment of the effect of a high-fat meal on the systemic exposure of the drug. The food effect information from such pilot studies can help with assessing the food requirements for subsequent trials. As formulation development continues, one can envision a scenario where the formulation that was used in the pilot study and the formulation that will be used in the pivotal safety and efficacy trials are significantly different. For such a scenario, the sponsor should assess the effect of food on the formulation to be used in the pivotal safety and efficacy trials to determine dosing instructions with respect to food prior to conducting the pivotal safety and efficacy trials. Another situation one may come across is that changes are made to the formulation used in the pivotal safety and efficacy trials after the trials are completed, and the formulation resulting from these changes is intended to be the to-be marketed or TBM formulation. A question often comes up is whether another food effect assessment needs to be conducted with this TBM formulation. The way this scenario has been addressed in the food effect guidance is that if the clinical trial formulation is significantly different from the to-be marketed formulation, a relative bioavailability study under fasting conditions and a food effect assessment on the to-be marketed formulation is recommended. On the other hand, if the clinical trial formulation did not show a food effect and the to-be marketed formulation is not significantly different from the clinical trial formulation, another food effect trial with the TBM may not be needed. Further, In case where a biowaiver is accepted to support a formulation change, the guidance indicates that a food effect study with the TBM formulation might not be necessary. Another important consideration is related to a scenario where efficacy or safety of a new drug is adversely impacted by food. Now, in such a case, the overnight 8 to 10-hour fasting may not always be practical to implement in real life, especially if the drug is given multiple times a day. So therefore, what is needed is a practical set of instructions so that the drug can be conveniently administered and still be safe and effective. In one of the subsequent slides, we will discuss how the food effect guidance specifically addresses this scenario and highlights the importance of assessing a practical time interval between drug administration and food. So far, we have discussed the importance of assessing the effect of food on drugs in order to determine whether food can impact the safety or efficacy of the drug. Various mechanisms by which food may impact bioavailability, and discussed a few key recommendations such as the importance of an early assessment of food effect using a high-fat meal, and if needed, a low-fat meal, and the concept of a pilot study. In the next part of the presentation, let us look into some general considerations for designing food effect studies. In terms of the trial design for a definitive food effect study, the food effect guidance recommends a randomized single-dose, 2 treatments, 2-period crossover trial. The 2 treatments correspond to drug administration under fed and fasted conditions. The guidance also outlines an option of using a 3-way crossover trial design in case the relative bioavailability of the formulations is also being assessed in the same trial, and I have included a reference to the FDA guidance on bioavailability studies submitted in NDAs and INDs for further information. For drugs with long half-life, and these drugs are defined as the drugs having a half-life greater than 24 hours in the food effect guidance, a crossover trial design approach will likely be challenging because of the long washout period. Therefore, the food effect guidance recommends a single parallel design for such drugs. In terms of the sample size for the definitive food effect trial, be it a crossover trial design or a parallel design, the food effect guidance does not recommend a specific number of subjects because the sample size will primarily be governed by the pharmacokinetic variability of the drug. Instead, the guidance provides a general recommendation to enroll an adequate number of subjects to sufficiently characterize the effect of food on the pharmacokinetics of the drug. Of note, the pilot food effect assessment that we previously discussed can be helpful to characterize the pharmacokinetic variability and inform the sample size. The food effect guidance recommends that for all orally administered drugs under development, the effect of a high-fat meal should be initially evaluated. If the results of such an assessment show a significant increase or decrease in exposure of the drug, the sponsor may consider evaluating the effects of a low-fat meal on the systemic exposure of the drug. In addition, conducting a food effect trial with a low-fat meal may be considered when administering the drug with a high-fat meal results in significant increase in toxicity or loss of efficacy, or if there are tolerability issues associated with administering the drug with a high-fat meal in the indicated patient population. Because we have been discussing the different types of meals that may be used for food effect assessment, let us review the caloric and fat composition of these meals. The guidance defines a high-fat meal as a meal containing 800 to 1,000 kilocalories, 500 to 600 kilocalories are derived from fat, which roughly corresponds to 55 to 65 grams of fat. The guidance defines a low-fat meal as a meal containing 400 to 500 kilocalories. 100 to 125 kilocalories are derived from fat, which roughly corresponds to 11 to 14 grams of fat. The guidance recommends that the sponsor should provide a description of the meal as well as the caloric content breakdown of carbohydrates, protein, and fat in the study report. Data collected on the type of fat, for example, percent saturated fat and percent unsaturated fat, can also be helpful. Examples of high and low fat meals are provided in the appendices of the food effect guidance and can help guide the study design and product labeling. In terms of subject selection, food effect assessments are generally conducted in healthy subjects rather than patients to minimize potential influence of any disease-specific factors. The underlying assumption here is that the effect of food on the pharmacokinetics of the drug will be similar in healthy subjects and patients. That being said, there may be instances in which food effect studies may be conducted in patients. For example, if safety concerns preclude the enrollment of healthy subjects, or if available information suggests differential food effects in the target patient population because of the underlying disease condition. The food effect guidance recommends that food effect studies enroll both male and female subjects unless the indication is specific to one sex, for example, oral contraceptives, or specific safety concerns preclude the enrollment of one sex. For example, if the drug is a teratogen, women of childbearing age should be excluded. The food effect guidance recommends that food effect studies should enroll subjects with normal renal and hepatic function, again to minimize the influence of confounding factors on the exposure of the drug in order to better characterize the food effect. Continuing with the theme of subject selection and efforts to minimize the effect of confounding factors, food effect studies should not enroll subjects who cannot refrain from using concomitant drugs that could confound the results of the food effect study. For example, drugs that can alter the absorption of other drugs by affecting gastrointestinal motility or by changing the gastric pH, or drugs that can increase or decrease the metabolism and excretion of the drug for which the food effect is being characterized. An important consideration for definitive food effect studies is the dose to be used. The guidance recommends that definitive food effect studies use the clinically recommended dose. There may be instances where several doses of a drug that exhibits linear pharmacokinetics will be marketed. In this case, the guidance recommends using the highest clinically recommended dose. That being said, if it is unsafe to administer the highest clinically recommended dose to healthy subjects, the guidance outlines two options. One, use the highest strength of the drug product in healthy subjects instead of the highest clinically recommended dose, or conduct the food effect study in patients. For drugs with nonlinear pharmacokinetics across the therapeutic dose range, the guidance recommends that single-dose food effect studies be conducted using both the high and the low doses. As far as drug administration goes, the food effect guidance describes the fasting and the fed conditions. When the drug is administered under fasting conditions, the drug should be administered to study subjects with 240 ml of water following an overnight fast of at least 10 hours. Additional water is permitted as often as desired, except for the period 1 hour before until 1 hour after administration of the drug product. The study subject should not consume any food for at least 4 hours after the dose. Subjects should receive standardized meals scheduled at the same time throughout the study. When the drug is administered under fed conditions, Subjects should start the recommended meal, be it a high-fat or a low-fat meal, 30 minutes before administration of the drug product and eat the meal within 30 minutes. The drug product should be administered to the study subjects with 240 ml, that is 8 fluid ounces of water. Additional water is permitted as often as desired, except for 1 hour before until 1 hour after administration of the drug product. The study subject should not consume any food for at least 4 hours after the dose. As I mentioned before, the food effect guidance recommends that for fasting conditions, the drug should be administered following an overnight fast of at least 10 hours. One may envision a scenario where fasted dosing is necessary because food can significantly increase or decrease the exposure of the drug. However, the standard overnight fasted condition might not be practical for patient treatment. Moreover, the results of the overnight fasted condition might not be applicable to shorter periods of fasting in patients. Therefore, to help with providing patient-friendly instructions in the prescribing information, for example, do not consume food within X hours before or Y hours after drug administration, the food effect guidance recommends that food effect studies with appropriate separation times between drug administration and food consumption should be conducted. The sponsor should provide the PK data to support pragmatic labeling instructions to prevent food-drug interactions after taking into consideration a variety of factors such as the frequency of dosing, the patient demographics, the disease condition, and any other relevant factors. In terms of sample collection for both fasted and fed treatment periods, the food effect guidance recommends that samples should be collected in a suitable biological matrix, for example, plasma from the study subjects. To characterize the complete concentration versus time profile for the drug, the total duration of sampling should cover at least 3 to 5 elimination half-lives of the drug. Of note, the sample collection times do not have to be the same for the fasted and fed treatment arm. If food is expected to alter the time course of drug concentrations, different sample collection times for the fasted and fed treatments can be used. To determine whether to measure other moieties in addition to the parent drug in the biological matrix, such as active metabolites, the food effect guidance recommends sponsors to refer to the FDA guidance entitled Bioavailability Studies submitted in NDAs or INDs, General Considerations. This brings us to the last segment of today's presentation, in which we will discuss a few other considerations related to food effect assessment. A question that often comes up is, can a food effect trial be waived? Based on all the available information so far, we know that in most cases, the systemic exposure of biopharmaceutics classification system class 1, that is BCS class 1 drug products, is unaffected by food. That being said, some BCS class 1 products are subject to high first pass metabolism effects or luminal degradation and can be affected by food. The food effect guidance recommends that a food effect trial may be waived for BCS class 1 products that also have a high bioavailability. The possibility of waiving a food effect trial is considered on a case-by-case basis. Hence, the food effect guidance recommends that the feasibility of obtaining a waiver for the food effect trial be discussed with the agency. I'm sure all of you are aware of the significant advancements that have been made in the area of model-informed drug development, especially with physiologically based pharmacokinetic or PBPK modeling and simulation approaches. The role of PBPK models to inform in vitro assessments that may be conducted to optimize parameters important for understanding food-drug interactions has been a subject of numerous discussions. Now, that being said, prospective prediction of food effect has been somewhat challenging, primarily because of the complex gastrointestinal physiology. PBPK modeling is a rapidly evolving tool and is an important component of the holistic approach to food effect assessment. The food effect guidance encourages the sponsors to reach out to the division to discuss the suitability of PBPK approach for food effect assessment. For drug products intended to be given with qualified soft food vehicles such as applesauce, pudding, etc., the soft food identification and qualification are conducted using in vitro methods. And this applies to all soft foods to be included in the label to demonstrate lack of potential physiochemical interaction between soft food and the drug product, and the food effect guidance refers to another FDA guidance outlined at the bottom of the slide. It is very important to note that in vitro assessments do not replace the need for an in vivo evaluation to permit instructions for drug administration with qualified soft food vehicles in labeling. For the labeling to indicate that the drug can be sprinkled on soft foods, the food effect guidance recommends that sponsors should perform additional in vivo relative bioavailability studies using the soft foods to be listed as vehicles in the proposed labeling. The labeling of certain products may recommend that the product be mixed with a liquid vehicle, for example, a beverage, before administration. For such cases, qualification of liquids for their use as vehicles should include an assessment and demonstration of lack of a potential interaction between the liquid and the drug product, either intact or manipulated, including interactions of the drug substance or excipients with the vehicle. For recommendations on in vitro assessments to select appropriate liquid vehicles for drug administration, the food effect guidance includes a reference to another FDA draft guidance entitled Use of Liquids and/or Soft Foods as Vehicles for Drug Administration, General Consideration for Selection and In Vitro Methods for Product Quality Assessments. Further, the food effect guidance recommends that sponsors should contact the FDA to determine what data should be submitted to support proposed labeling instructions regarding the use of specific liquids as qualified vehicles for drug administration. In terms of specific populations, the food effect guidance does not recommend that a separate food effect trial be conducted in geriatrics. For pediatrics, when a new age-appropriate pediatric formulation is developed, the food effect guidance recommends that the sponsor should conduct a new food effect study with the pediatric formulation in adults. These results can then be applied to the pediatric population. The food effect guidance recommends that sponsors can use foods and quantities of food that are commonly consumed with drugs in a particular pediatric population. The outcomes of food effect studies conducted with one soft food vehicle could be extrapolated to other similar soft food vehicles, for example, the same fat, protein, and carbohydrate content, and could support proposed labeling instructions. As previously mentioned, such extrapolation should be supported by in vitro data, which demonstrates a lack of interaction between the drug product, intact or manipulated, including lack of interaction with the drug substance or excipients with other similar soft food vehicles. Small quantities of liquids or soft food, for example, 5 to 15 ml, to be used as vehicles for pediatric drug delivery should adhere to the principle described in the FDA 2018 draft guidance entitled Use of Liquids and/or Soft Foods as Vehicles for Drug Administration, Journal Consideration for Selection, and In Vitro Methods for Product Quality Assessment. When a formulation that is approved for adults is approved for use in a pediatric population at least 12 years old, a separate food effect study is not necessary. A separate food effect study might not be necessary if the pediatric formulation is very similar to the adult formulation and if the pediatric formulation is approved based on a biowaiver approach. This slide discusses the food effect related considerations for fixed combination drug products. Fixed combination drug products are products with two or more active ingredients combined at a fixed dosage in a single dosage form. Fixed combination drug products can exhibit different effects of food compared to when each active ingredient is administered alone. The effect of food on the various active ingredients on the fixed combination drug product after administration of the fixed combination drug product should be assessed. Food effect study might not be necessary if neither drug product, when given alone, is affected by food, and the sponsor can provide an adequate justification that the fixed combination drug product would be unaffected by food. In terms of data analysis, the food effect guidance recommends that exposure measures and pharmacokinetic parameters, and the guidance outlines a detailed list of these pharmacokinetic parameters, be derived from the food effect studies and reported. Individual subject measurements as well as summary statistics, for example, group averages, standard deviations, coefficient of variation, ranges, should also be reported. Exposure measurements, AUC and Cmax, should be log transformed before statistical analysis. The 90% confidence interval for the ratio of the population geometric means between the fed and the fasted conditions should be provided for AUC 0 to infinity, AUC 0 to T, and Cmax. The clinical relevance of any differences in Tmax and Tlag should also be described. An absence of an effect of food on bioavailability is established if the 90% confidence interval for the ratio of the population geometric means between fed and fasted treatments constructed based on log transform data is contained in the equivalence limits of 80 to 125% for AUC 0 to infinity or AUC 0 to T when appropriate, and Cmax, unless other criteria based on the established exposure-response relationship for the drug are more appropriate. It is important to note that when the 90% confidence interval for the ratio of the population geometric means of either AUC 0 to infinity or AUC 0 to T when appropriate, or Cmax between fed and fasted treatment fails to meet the limits of 80 to 125%, the food effect guidance recommends that the sponsor should provide specific recommendations on the clinical significance of the effect of food based on what is known about the exposure-response relationships of the drug and the totality of the available information. In summary, although food effect trials are designed to assess the worst-case scenario, which is mostly the effect of a high-fat meal on the pharmacokinetics of a drug, it is important to also consider more clinically relevant scenarios such as other meal types and modified fasted conditions. It is critical to consider exposure-response relationships when interpreting the results of food effect trials. Providing clear and actionable recommendations in relation to food is important to facilitate the safe and effective use of drugs. The food effect guidance includes an appendix with a few examples of incorporating food effect information in the label. Now let us try to address two challenge questions based on the information that was presented in the webinar. Challenge question number 1. Low-fat meal as defined in the food effect guidance contains how many total kilocalories? A, 400 to 500. B, 500 to 600. C, 800 to 1000. And D, 1000 to 1200. The correct answer is A, 400 to 500. The second challenge question is, food effect guidance recommends evaluating food effect using the following type of meals. A, high-fat meal only. B, low-fat meal only. C, high-fat meal and low-fat meal as needed. And D, none of the above. The correct answer is C, high-fat meal and low-fat meal as needed. This brings us to the end of the webinar. Thank you very much for your time and attention.
Thank you for that great presentation. We will now transition into our question and answer session. If you have not had a chance to enter your questions in the question and answer chat pod, please do so now. We will answer as many questions as time allows. So let's see, we have our speaker and our two panelists to help answer questions. So let's start with a question for Dr. Arya. Dr. Arya, what considerations can the agency share regarding the design of a relative bioavailability study to compare the exposure when taking the drug with a low-fat meal versus a high-fat meal for a drug that exhibits increased exposure in the presence of a high-fat meal?
Thank you for that question. So if it has been already established that the exposures of the drug increases in the presence of a high fat meal, and there is interest in assessing the effect of a low fat meal on the same formulation, then a randomized balanced single dose two treatment fed and fasted 2 period crossover design should generally be used. And the low fat meal will correspond to the fed treatment arm in this trial. Now, if the goal is to assess the effect of both a high fat meal and a low fat meal in the same trial, then the trial design I just described can be expanded to a three-way crossover trial where the three treatments, namely the fasting, low fat, and high fat meal, can be evaluated. Thank you.
Thank you, Dr. Arya. We have another question for you. For drugs with time-dependent PK, such as autoinducers, would you recommend for a single dose and multiple dose design? If multiple dose, would you expect to administer with food daily until SS?
Yes, thank you for that question. So, for drugs with non-linear pharmacokinetics across the therapeutic dose range, the food effect guidance recommends that sponsors should conduct a single dose food effect study using both the high and the low doses listed in the proposed product labeling. Thank you.
Thank you. And one more question for you. At what phase of small molecule drug clinical development should the food effect study be conducted?
Yeah, thank you for the question. So the food effect guidance recommends that the effect of food be initially assessed as part of the first in human single dose trial, also referred to as the pilot food effect assessment in the guidance, to provide a preliminary assessment of the effect of a high fat meal on the systemic exposure of the drug. And if the formulation that was used in the pilot study and the formulation that will be used in the pivotal safety and efficacy trial are significantly different, then the sponsor should also assess the effect of food on the formulation to be used in the pivotal safety and efficacy trials to determine dosing instructions with respect to food prior to conducting the pivotal safety and efficacy trials. Thank you.
Thanks. Let's move on to Dr. Booth. Dr. Booth, what percent of food effect on the drug bioavailability suggests a recommendation for taking the study drug with food or 30 minutes after the meal?
Good afternoon. Thank you for the question. So with food effect studies, it's not like bioequivalent studies. We don't really have no effect boundaries. Okay, so for each product, it's really based on what you know about the exposure response relationships for efficacy and safety. So for instance, you might have a drug with a very narrow therapeutic index, and in that sort of situation, food
Sorry for the technical difficulties. We'll move on to some questions for Dr. Stier in the meantime. Dr. Stier, is there a difference in the design of a Fed study for 505B1 versus 505B2?
Thank you for the question. No, the design of a food effect study will be the same for both 505b1 and 505b2. Thank you.
Thank you, Dr. Stier. We have a couple more questions for you. Next question, do we need to conduct food effect studies for 505b2 products?
Thank you for the question. Do we need to conduct food effect studies for 505b2 products? Yes, as outlined in the guidance, bioavailability studies submitted in the NDAs or INDs, food effect studies on new drug products, including 505b2, should be conducted to evaluate the effect of food on BA. You can also please refer to the recent SDI webinar on YouTube for more information. Thank you.
Thank you, Dr. Stier. We have one more question that came in for you. Does the draft M13 guidance in BE for IR products discuss food effect studies for new drug development?
Thank you for the question. So in regard to the draft M13 guidance for BE for IR products and its potential impact on food effect studies for new drug development, just the first thing I want to do is stress that the M13 is currently in draft form and has not been finalized. So any discussion here is just preliminary, and I certainly encourage everyone to keep track of the guidance and look for the finalized form down the road. But in the current draft version, food effect studies conducted during new drug development are carved out or exempted from M13. So if you're doing a food effect study, you want to refer to the guidance we're talking about today, as well as the bioavailability guidance as well. If the audience is interested about some of the other information, which is outside of the scope today, I'll also refer you to another recent SDI webinar on the M13 that's posted on YouTube. Thank you for the question.
Thank you, Dr. Stier. Let's try going back to Dr. Booth. Dr. Booth, back to that question. What % of food effect on the drug bioavailability suggests a recommendation for taking the studied drug with food or 30 minutes after the meal?
Thank you. Sorry for technical difficulties. As I was saying, there are really no fixed effect boundaries for food effect studies. It really depends on your understanding of the exposure-response relationships for efficacy and safety. And as I was saying, for a narrow therapeutic index drug, a small change in exposure, say 20%, might be something that's of concern. So you'd have to do something about that with how you want to dose the product with respect to food. But in other cases where the therapeutic window is much wider, you could, have a larger effect on the exposure, and that may not really necessitate any action. And so you might give that drug with or without respect to food. Thank you.
Thanks, Dr. Booth, for that answer. We have another question for you. Does FDA have a definition of long half-life, like 24 hours as the cutoff point?
Yes, we do. And actually, it is 24 hours. So anything with a half-life of 24 hours or longer is considered a long-acting drug. We mentioned that in the food effect guidance. And in those kind of cases, you might want to look at a parallel design kind of study rather than a crossover design in order to deal with those longer sampling periods. Thank you.
Okay, Dr. Booth, one more for you. Have you seen drugs taken with food have had less serious or no GI issues than the same drug taken without food?
Yes, we have. Actually, Zicadia, suritinib, is a very good example of that. That was a drug that was approved to be administered in the fasted condition originally, but it turned out it had a lot of GI toxicity. And so the company went back and did some food effect studies with a lower dose of the drug and determined that at this lower dose, they were able to match the systemic exposure of the higher dose in the fasted state. But by giving it in the fed condition, they reduce GI toxicities very considerably. And so the labeling now indicates taking Zicadia with food at this lower dose to treat patients. Thank you.
Thanks for that response. Let's go back to Dr. Arya. Dr. Arya, is high fat and high calorie, are these terms interchangeable? Okay, it looks like we're still experiencing some technical difficulties. So Dr. Booth, back to you. We have a few questions that have come in for you. Are there any examples of food effect studies incorporated in phase 1 oncology trials, especially in
Okay, thank you for that question. So yes, there are, in fact, we do see food effect studies in oncology trials fairly frequently. So generally, dose escalation steps are conducted in fasted patients, and often the sponsor will include a fed cohort in one of the intermediate doses to get some sense of whether or not there's a food effect in order to make decisions about how to administer the drug as they move forward. Then later in development, you would generally see a food effect study with the final formulation. We have also seen cases where dose escalation was done in fed patients. And then partway through the dose escalation, they've actually incorporated a cohort of patients who were fasted. So again, so you can get a sense of the impact of food on the pharmacokinetics of the drug. And another variation that we've seen is that at the completion of the dose escalation, when you select the recommended phase 2 dose, often in oncology, there's an expansion at that phase 2 dose to get a better sense of the activity and the safety profile of the drug. And in that setting, we have seen examples where in the course of doing that part of the study, they've also incorporated a cohort of patients who were fed, so you could see what the impact of the food was. So a number of designs are possible. And for those of you folks who are interested, I know a number of these are published, so they're out in the scientific literature if you want to read up on those more. Thank you.
Thank you for that response. Excuse me. Another question for you. When is it necessary to establish efficacy in a new formulation that removes the food effect?
Yeah, that's a good question too. So generally, if you have a formulation with a food effect that behaves pharmacokinetically significantly differently from the reference product under the conditions of use in the indicated patient population, then you may need to assess either the efficacy or the safety or both in some fashion. The Janssen formulation of abiraterone is an example of this. Okay, it behaved quite differently from Zytiga, and so they had to do some additional studies for that. Thank you.
Thank you, Dr. Booth. One more question for you. It just came in. How do you account for disease impact on food digestion or absorption in cancer patients?
Thank you for that question. So this can be a very difficult issue. We have seen cases where the pharmacokinetics of the drug and the food effects have been determined in the healthy volunteer population, and then they've determined the pharmacokinetics in one or more oncology populations. Okay, so sometimes there are no differences between the healthy volunteers and the patients, and you can just extrapolate the food effect finding from the healthy volunteers to the oncology patients. But we've also seen examples where the pharmacokinetics have been significantly different between the healthy volunteer patient population and the oncology patients. Okay, so what does this mean when it comes to trying to interpret or extrapolate the food effects from the healthy volunteer study to oncology patients? That can be very difficult. Unfortunately, currently the science supporting PBPK has not really matured enough yet for us to make this a useful modality for exploring or understanding these issues in silico. And so really the best course is when at all possible, you should conduct a food effect study in oncology patients. This is probably especially true for GI cancers. Thank you.
Thank you, Dr. Booth. Let's go back to Dr. Arya. Hopefully the technical issues have been fixed. Dr. Arya, is it mandatory to perform a pilot?
Thank you for that question. No, it is not mandatory, but recommended. And a pilot study can provide a preliminary assessment of the effect of a high fat meal on the systemic exposure of the drug, and the food effect information from such a pilot study can help with assessing the food requirements for subsequent trials. Thank you.
Thank you, Dr. Arya. We have another question for you. If the product label recommends more than one soft food, is it required to demonstrate the food effect with all listed soft foods, or any one of them is adequate as part of the site change or formula change kind of situations?
That is a really interesting question. So the soft food identification and qualifications are all conducted using in vitro methods, as I described in the slides. Uh, though these in vitro assessments do not replace the need for an in vivo evaluation to permit instructions for drug administration with qualified soft food vehicles and labeling. And for the labeling to indicate that a drug can be sprinkled on soft foods, the food effect guidance recommends that the sponsor should perform additional in vivo relative bioavailability studies using the soft foods to be listed as vehicles in the proposed labeling. Thank you.
Thanks. And we just have a little bit left. Let's try to squeeze one more in for Dr. Stier. Dr. Stier, for a 505(b)(2) NDA submission for a modified release product, since the food effect already is done for the existing product, will the new MR product or other type of formulation under 505(b)(2) still have to conduct all of the required FE studies? It looks like we're having audio issues again. I apologize. But it looks like that's all the time we have for questions today. Thank you to our presenters for the great presentations and for answering all these questions that came in today. And thank you to the audience for asking the questions.